that we have are these Galeria melanella. They're the wax moth.
Oh yeah.
And we're using those for a slightly different set of experiments because they're very good. Whereas fruit flies are very good to understand the kind of basic biology, cell biology, how cells work and function. The wax moths are a much better model for immunity and infection. So most insects, as I say, kind of grow anywhere happily between 20 and 25 degrees.
Yeah.
These wax moths, their optimum temperature is sort of 33.
Oh wow.
32, because they're a pest of beehives.
Oh.
Actually, the temperature of a beehive is around about 30 to 32
Yeah,
degrees.
that's how they kill invaders, isn't it? They kind of envelop them and then flap and overheat them. Yeah,
absolutely.
yeah.
But so what that means is that they're also quite happy at 37 degrees, and 37 degrees is our body temperature. So if you're trying to find a model organism where you can study the infection dynamics of a particular pathogen, like a nasty bacterium or something, it's not ideal to be using a model that grows at 20 degrees because the bacteria themselves will be growing at our body temperature.
Yeah.
So we can use those wax moths as an alternative to using mice and rats for experiments.
Yeah.
And use those to study infection and immunity.
Yeah.
And to be honest, most people, there are exceptions, most people don't have a real problem with experimenting on maggots.
No.
But they do have a problem with experimenting on rodents and mice and rats and things like that. So actually, the more experiments that you can do using an insect model, the smaller the number of experiments that you have to do on mice and rats. Is the way we look at it.
That's a very good, valid point. Yeah.
So our moths. So this is the kind of modified example, similar to the flies. We have a source of food. In which case this is a food which is a mixture. Again, it's a mixture of a carbohydrate source like a maize and a flour, but this one has honey in it because obviously they like honey. They come from the hives. So you can see there are the larvae. So they're much bigger than the fruit fly, and actually these are the smaller larvae. So they get to be a little bit bigger than that even. So they'll lay them.
There are tiny little things also.
These little dots down there.
The little mites that are... oh no, that's just their heads.
That's their heads.
Oh, I saw it as a separate creature. Isn't that weird?
They're there. So they're wandering along all like that.
Yeah.
They're quite happily away.
Yeah, they look very happy actually, very busy.
Yep, so they're busy on that, and they're even spinning silk. Can you see at the top there?
Oh yes, I can.
Their little silky webs. So some of those will be getting ready to pupate. So then they go from there. Have they got any pupae in there? So you can see they get a little bit bigger. So these ones do have little prolegs.
Okay.
So they move quite quick as well.
Yeah.
These little ones. I will keep that in there. And then... Okay, no, so they're the injected ones. So we're making flies that moths that glow as well.
Oh, lovely.
Different experimental.
Yeah.